Hello everyone, a very warm welcome to all. I'm Apurva Bharadwaj. I'm currently working with Intrinsic Analytics as the business and market analyst. Today I'll be talking about the promiscuous nature of a protein, insulin-like growth factor binding protein 3, which is also the key protein for my graduate research work as a QE scholar in Winnipeg. So it is believed that science makes more sense when you know why you're doing it, more than how you're doing it. So before I get into the details of my research work, I really want to tell you why I think my research is significant. So my research provides novel insights into gene expression and also the regulation of a key growth pathway. Because my protein has a really long name, so for comfort and ease, throughout my talk, I'll be referring to it as IGF-BP3. So IGF-BPs, they're highly conserved proteins that are secretory in nature, which means that they're formed and they're transported outside the cell. They are hepatic in origin and they also function as a transport vehicle for the IGFs. Now let me take a step back and introduce you to the IGF family. The IGF family comprises of three major components. The IGFs, that's the insulin-like growth factors, which mediate cellular processes such as cell growth, cell survival, and cell death. The IGF-BPs, that are the insulin-like growth factor binding proteins, which is what we'll be talking about in a bit, and IGFRs, which is insulin-like growth factor receptors. As the name suggests, they're the receptors for the factors. Now, my research was focused on the role of IGF-BP3, which is also the third most abundant protein expressed in human bones. Now, before I dive into details, let me give you a little more information about IGF-BP3. So, IGF-BP3 is a hepatic protein. It is secretory in nature. It has been found to be expressed in the cytosol, inside the nucleus, and even within the blood serum. It is one amongst the 6 members of the IGF-BP family. It is considered as a key protein that makes IGFs available to the IGFRs. That means it regulates the availability of these factors to its receptors. Now, IGF-BP3 has been found to function via 2 major pathways. One is the IGF-dependent pathway and the other is the IGF-independent pathway. Now, in the IGF-dependent pathway, the IGF complexes with the IGFR, and together as a complex, they help and mediate the cellular processes such as differentiation, metabolic activity, etc. Whereas in the IGF-independent pathway, IGF-BP3 no longer requires IGF or IGFR to mediate its functions. Also, here, the cellular processes that the pathway regulates or mediates are anti-proliferative activities, which clearly means it's completely the opposite of what it does in the IGF-dependent pathway. A significant amount of literature outlines the interactive nature of IGF-BP3. So what that means is that this protein has been found to have interactions with nuclear proteins and nuclear receptors. Also, proteins that have been found to translocate into the nucleus have been found to play an important role in transcription or even regulate the process of gene transcription. Now, considering this literature which was available, we were very much interested and intrigued to investigate the potential nuclear binding proteins that could be interacting with IGF-BP3. And we also wanted to understand what makes IGF-BP3 translocate into the nucleus. This paved way for my very first objective, which was to determine whether nuclear IGF-BP3 interacts with histone protein. Now, what are histone proteins? Histone proteins are highly alkaline proteins. They are present in eukaryotic nucleus and they help to package the DNA in a superstructural subunits called nucleosomes. These proteins are indeed the ones around which the DNA coils and therefore they play a super important role in gene regulation. Now, in order to answer the objective, I carried out an experiment which was called the ligand blot experiment. Now, what I did here was I took two cell lines. One was a human liver cancer cell line, which is the HepG2 cell line. I grew them, and I also took the mouse myoblast cell line, which was the C2C12 cell line. I grew those plates to a confluency. I fractionated the cells into a cytoplasmic and a nuclear fraction. I enriched the nuclear fraction into further subfractions, one which was highly concentrated with DNA and histone, and the other that had transcription factors. Now, once I had these fractions, I resolved these fractions on an SDS page, which is a technique to separate the proteins according to their molecular weight. Now, once the proteins were separated, the gel was run. I transferred the proteins onto a membrane and incubated that membrane with the IGF-BP3 protein itself. Now, essentially, why I did this was so that the IGF-BP3 gets to interact with all the potential partners on that particular membrane. And indeed, that did happen. So it was a win-win situation. In order to see the blot, I used a chemical reagent to develop the blot and saw bands at certain areas. Now, those bands clearly suggested that IGF-BP3 did interact with some of the nuclear proteins. Now, because it was a SDS gel, we could clearly see the different molecular weights of the proteins which were interacting with IGF-BP3. And surprisingly, one of them was histone 3, which was 17 kilodaltons. This clearly suggested that yes, IGF-BP3 does interact with histone protein, which was what we were looking for. So with this, we were the first ones to report that nuclear IGF-BP3 does interact with histone 3. And this also had a direct implication, which was also the overreaching goal of my research, which clearly suggested that the interaction of a key growth pathway protein, which is IGF-BP3, with histones could have a significant and a direct effect on chromatin modeling and therefore gene regulation. With this, I would like to thank Soapbox Science once again for giving me this opportunity to talk about my research. My due reverence to my supervisors, Dr. Shelly Verma Srivastav and Dr. Anurag Srivastav, the funding organizations, my family and friends, and not the least to the Queen Elizabeth Scholarship Trust for making this possible. Thank you so much.
Hi, I'm Shreya Dhume, and I'm a PhD candidate in the University of Manitoba. My research interest is neuroscience, and in neuroscience, I study autism. But first, let's start with this guy. He is one of my favorite toys from my childhood, and I bought him because I wanted to learn how the human body works. And he was fantastic. He told me what all the organs are, how they work, and what controls them. And what I found was that the human brain is actually the mastermind that controls all the organs. And that's why I wanted to learn neuroscience and how the brain works. So the human brain is around 1 to 2 kgs in adults, but for a small size, it usually takes up most of the energy compared to all the organs. And this is because it controls everything that happens in your body. It controls how you breathe, how the heart beats, how you walk, how you talk. It even forms memories. So how is that the small organ does all this? So let's dive in deeper. But first, let me give you an example. Imagine you touch a pot of boiling water. What the first thing you do is you retract your hand. You withdraw it because it's so hot. But in your body, what is the sequence that actually happens? First, there are specialized cells in the skin which sense the really hot temperature. And then it transmits all this information to the brain. Now the brain is a wonderful thing. It computes everything, your sensory information, your emotions, that you were scared while touching it, it was really hot. It computes all this and then sends the output to the arm muscles to withdraw your hand. Now you might think this is a big sequence and it requires a lot of computing, but it happens within milliseconds. And why is that? Because brain primarily works through electrical activity. So you might wonder what this electrical activity is. The electrical activity happens because of a specialized cell in the brain known as a neuron or a nerve cell. Now this is actually the longest cell, one of the longest cells that are present in the body. It extends, for example, from your back till your feet, and it transmits all this information directly to the brain and computes everything. So like us, neurons are social creatures. They need to talk to each other, they need to communicate. So how do they do that? For example, here is neuron 1 and it wants to talk to this neuron. So how does it do? It usually sends chemical transmitters and talk to each other. But this happens at a specialized place known as a synapse. The synapse is just a 20 nanometer very small gap between two neurons. So this is how they talk, through a synapse. Now in the brain, there are millions of neurons. And each neuron have millions of synapses. And they all talk to each other through this gateway. Now as I said before, the synapse is a gap of around 20 nanometer wide. But is it actually empty? No. It is filled with proteins known as cell adhesion proteins. Now why is it called adhesion? Because these proteins actually bridge together two neurons. They help in communication and they do one more other thing. They give identity to the neurons. Now what is this identity? Now there are two types of neurons or synapses. There's an excitatory synapse which promotes the electrical activity, and there's inhibitory which suppresses this activity. And a balance between these two gateways or these two neurons is very crucial for the proper functioning of the brain. Now imagine a tilt towards more excitatory or more inhibitory. This is what exactly causes improper brain function, and this imbalance is the hallmark of the neuropsychiatric disorders. My research project includes studying one of these proteins known as the synapse organizing proteins, and that is exactly what they do. They organize the synapses, but they also maintain the balance that I just talked about the inhibitory and the excitatory synapses. Now, there were numerous studies where changes in these proteins can lead to neuropsychiatric disorders like autism, schizophrenia, intellectual disability, and even suicide risk. My research focuses on autism. In Canada, 1 out of 66 children are diagnosed with autism. Most of the research in the field of autism is trying to understand how changes in this gene can increase the risk of autism and also find out what is the mechanism behind it. And this is exactly what I do. I study these synapse organizing proteins, but I study them in mouse models. Now, why mouse models? The mouse brain is genetically similar to the human brain, and it also functions almost similar way. Another main reason is that you can genetically manipulate mice, and it's easier to do behavioral studies on them. To give you an overview of my study, I genetically manipulate the synapse organizing proteins in mice. I do this by either deleting them or making changes in them. Then I study what happens to the neurons in the brain. And I do this using 3 criteria. The first one is what about the neuron health? Whether they are happy or they are sad or are they barely surviving. Next is the electrical activity, whether it's increased or decreased. And this will tell me whether there's an imbalance in the brain. The third one is changes in the behavior. This will tell me how the imbalance is causing what types of behaviors. And what I found out was when I deleted my protein of interest, there was decreased activity in all 3 of it before birth, after birth, and also in the adult stage. And this is very important because most of the results of my study correlate to the impacts we see in autism. So what does this mean? This means that the protein of my interest, that is the synapse organizing molecules, actually regulate how memories are encoded and thus affects the behavior of an individual. The treatments available right now for autism are limited to just treating the symptoms with speech and behavioral therapies. However, future research hopes to find new therapies as we study more and more about these critical proteins and their functions in the brain. And restoring the proper function of these proteins can help restore the balance and thus bring us one step closer to solving these complex disorders. So this is all about my research. I hope you guys enjoyed this talk as much as I enjoyed presenting it. Thank you guys for listening and have a good day. Thank you.
Hello, my name is Silvia Leon and I am a physician by training and a data scientist here in Winnipeg, Manitoba. I work using large health databases to evaluate outcomes in patients with chronic kidney disease. Today, I want to talk about my research in hyperkalemia and kidney disease. What is hyperkalemia? So, a hyperkalemia is a high potassium blood levels. Potassium is a mineral and one of the body electrolytes. Potassium is involved in maintaining normal blood pressure, transporting nutrients into the cells, and support healthy nerves and muscles. Your heart is a muscle, so potassium also helps with your heart rate. So potassium is found in several foods, especially fruits and vegetables. The most known one for potassium is the bananas. So because potassium works closely with your muscles and your nerves, having high levels or low levels of potassium can be dangerous. Many people don't experience symptoms of hyperkalemia. If people experience symptoms, they might have tiredness, muscle cramps, muscle weakness, chest pain, or difficulty breathing. If the potassium levels increase acutely, or if they increase very high, they might even cause a heart attack or death. The potassium levels are measured in a blood test. The test is a blood test like the ones that you are familiar with. If your potassium levels are higher or equal to 5.5 millimolar, you are having hyperkalemia. So what is the relationship between kidney disease and potassium? Well, the kidneys help your body to excrete the excess potassium. When you have kidney disease, your kidneys cannot remove adequately that extra potassium and it starts to accumulate in your body. 1 out of 10 adult Canadians have kidney disease. That is about 4 million Canadians with kidney disease. More than half of the patients with kidney disease would have hyperkalemia. And hyperkalemia is one of the most common electrolyte disorders in patients with kidney disease. Besides kidney disease, other risk factors for hyperkalemia are heart failure, diabetes, and medications that we use to treat heart failure, diabetes, hypertension, and kidney disease. After an episode of hyperkalemia in patients with kidney disease, the risk for mortality increases. So controlling these high potassium levels is essential for reducing the mortality risk in this population. The choice of treatment is determined by several factors, like how rapid the potassium levels increase, how high are those potassium levels, and if the patient is having or not changes in the electrocardiogram. The emergency treatment is needed if the potassium increases very fast, if it's very high, or if you're having changes in the electrocardiogram. The treatment could consist of putting calcium through an intravenously, or giving you glucose and insulin also intravenously, that will lower the potassium while the doctors manage to control what is causing that high potassium. Besides emergency treatment, there are other strategies that the doctor might recommend you to decrease the potassium levels or keep them low. One of them is the dietary management. As I mentioned before, we have potassium in fruits and vegetables. So some of the medications you need to avoid, some of the fruits and vegetables you need to avoid are bananas, avocados, asparagus, and some dried fruits. Ask your healthcare provider if you are at higher risk of hyperkalemia and what will be the ideal amount of potassium for you. Some people may also need medication that will help them remove the extra potassium in their body and prevent that potassium levels to go back again. So here's what I want to study. These patients with kidney disease very often are prescribed a type of medication called renin and angiotensin-aldosterone system inhibitors, or RAS inhibitors. These medications are used for the treatment of kidney disease because they have shown to reduce the progression of kidney disease, to improve cardiovascular disease, to improve the heart function, and to decrease the risk of mortality in these patients. However, they are also associated with high risk of hyperkalemia. So here is the dilemma. The treatment with RAS inhibitors is often modified or discontinued once a patient has a hyperkalemia event. So this might lead to a greater threat of kidney disease and heart failure. So to address this dilemma, we are conducting a large population study where we are going to examine in those patients who had hyperkalemia and discontinue the medications or lower the medication, what happened in terms of outcomes like death and cardiovascular events compared to those patients who continue the medication or continue to recommend the RAS inhibitors. To do so, we are using large anonymized health data. This health data is the one that us as data scientists use. This data contains all the medical information of the patient, like hospitalizations, surgeries, medical visits, diseases, every prescription, and every lab that the patient had. I organize, analyze, and understand this data using statistical analysis. By crunching numbers, we as data scientists are finding ways to understand human disease and to transform it into things that are policy relevant and something that people can understand. Manitoba has one of the most complete health datasets. On this project, I also work with nephrologists and other data scientists from the University of Manitoba, and it's a lot of fun to work with them. So I will do a simplified explanation of what I'm doing with the data. So I have all this large data set and I need in a time frame all the patients with a diagnosis of kidney disease. And among them, who are receiving the prescription of interest for my study, in my case, the RAS inhibitors. I can link to these patients other important information like age, other diseases, other prescriptions that they are receiving, and then I will see the laboratory data. And I can see in those who had a potassium tested if they have experienced hyperkalemia, if they have a potassium higher or equal than 5.5. Once I have this will be my population of interest. As I mentioned before, I also have data on the prescription, the medications that the patients are receiving. So I can see after that episode of hyperkalemia what happened with the RAS inhibitors. If they continue, discontinue, or lower the dose of the medications. Because this is real life data, we know that these decisions of treatment are always changes with the patients, are always changing with the patients. So some patients with their healthcare providers, they will decide to discontinue forever the medication, so others will continue as it is. There may be some that that continue the medication but at lower dose than the recommended ones, and there will be others who will alternate between continuation and discontinuation of the medications. And I will follow all those changes during my study. So we will form a set of estimates that will allow us to rank those who continue versus those who discontinue. And in a common outcome, I will be able to see in those who had a higher rate of adverse events. This work is important beyond our lab. For one, potential discoveries can help patients and doctors making healthcare decisions. They can teach them how to best approach the treatment after an episode of hyperkalemia. It also has space for policy intervention, as it's showing that there is a need for therapies for chronic hyperkalemia in patients with kidney disease. Lately, 2 new medications are being studied for the treatment of chronic hyperkalemia. So far, these medications in short-term studies have shown good results in treating hyperkalemia. And they have doing so while the patients are maintaining their treatment with RAS inhibitors. We are still expecting the results for long-term effects of these medications. So there is a need to evaluate the benefits of maintaining and optimizing RAS inhibitors despite hyperkalemia in patients with kidney disease. And that's my study. Thank you.
Hello, good day everyone. I am Rumana Islam and welcome to the Soapbox event today. I will share some of my research findings with you involving production of biofuels from cellulosic materials. If I ask you a question, what do you think are the top 3 global crises today? Let's see if your answer matches my thoughts. I believe none of you will disagree with me that accumulation of waste is a real problem. Therefore, reduce, reuse, and recycle is more important than ever. And the impact of petroleum industries on global climate change due to the greenhouse gas emission is enormous. And we must find some cleaner alternatives. Moreover, the reserve of fossil fuel is finite and demands for renewable energy sources are rising continuously. Through my research, I attempted to address these issues. Cellulose-rich waste are being generated by all sorts of human activities such as municipality waste, residues from agriculture and farming, industrial waste, think about pulp and paper mill. All these waste streams are rich in plant biomass, termed as lignocellulose. Transformation of lignocellulose into value-added chemicals such as ethanol and hydrogen offers attractive solution to critical problems world is facing now. Ethanol is already well known as an alternative fuel to most of us. Currently, fuel ethanol is basically made up from corn or wheat, which are both food crops. Also, biological hydrogen derived from non-food residues originated from agro-industrial and forestry activity is a promising energy carrier for future. So my main aim was to replace grain-based biofuels with cellulosic biofuels, which will eliminate the competition between food and fuel, at the same time addressing the issues of greenhouse gas and waste accumulation. This image shows a complex structure of lignocellulosic material present in plant cell walls. As you can see, it has 3 different types of polymers: cellulose, hemicellulose, and lignin. And to free these cellulose fibers from this strong structure, we need multi-step processing, which includes harsh mechanical, chemical, and thermal treatment. Solubilization of solid cellulose materials is required before fermentation. Cellulose makes up to 50% of plant dry mass, which is basically chains of many repeating sugar molecules. These sugars can be converted to bioproducts, which are cleaner and sustainable sources for biochemicals and energy. A good number of different fungi and bacterial species are capable of direct fermentation of cellulose into bioproducts. They accomplish it with a set of cellulose degrading enzymes called cellulases. We selected a smart species of bacterium named Clostridium thermocelum, which is so far the most efficient cellulolytic bacterium. This bug is equipped with a powerful set of cellulase enzyme systems called cellulosome. Just like a one-pot magic recipe, which is known as consolidated bioprocessing, it combines all important steps into one vessel. Production of multi-enzyme cellulose complex, hydrolysis of cellulosic solids, and simultaneous fermentation of released sugars. This eliminates the very expensive step for enzyme production and saccharification. In nature, biological reactions occur spontaneously, but in research laboratories, a controlled environment is created inside bioreactors to support these bioconversions. As you can see here, a reactor vessel that holds the growth medium, which is composed of nutrients essential for microbial growth. Common ingredients of growth medium are one or more carbon sources, vitamins, minerals, and salts. Inside this vessel, temperature, pressure, and nutrients and product levels can be controlled to mimic the natural environment of the selected microbe. In our case, Clostridium thermocelum loves oxygen-free atmosphere, so aeration cost is saved. However, we need nitrogen spurging to keep the oxygen from coming in. Composition of nutrients medium that supports growth and metabolism of microbes is a crucial determinator for rates of production of ethanol and hydrogen. First, I screened and selected the most influential nutrients. And there I found that concentration of cellulose, yeast extract, and magnesium chloride are the most important ones. Then I took them for further investigation to see how varied concentration of these nutrients affects biofuel production during direct conversion of cellulose by Clostridium thermocelum. I applied the response surface methodology to study the combinatorial effects of these key nutrients. And this picture shows the final outcomes of response surface methodology analysis. With varied concentration of yeast extract and cellulose, hydrogen concentration changed. The optimum condition predicted for hydrogen resulted about 3.2-fold more hydrogen compared with the unaltered medium. Same ways, ethanol production rate also doubled in the optimized medium. These are the significant improvements over the non-optimized version of growth medium. Now, these lab scale results looks very attractive. However, they need to be verified with scaled up conditions. Cellulosic biofuels hold great potentials to replace fossil fuels for some unique benefits associated with them. These are higher octane numbers of ethanol, which can be up to 100, whereas gasoline has an octane number close to 85. Hydrogen burns clean in fuel cells, resulting only water as waste product. Greenhouse gas emission is reduced by more than 85% for cellulosic bioethanol. Both of them uses renewable and low-cost raw materials and results solid waste minimization. and refining efficiency is same or better than gasoline. Despite all these promises, commercial scale production of cellulosic biofuel sector is progressing very slowly. Only about 20 commercial scale cellulosic ethanol plants were constructed in Europe, US, Norway, Brazil, China over the last decade. However, most plants are non-operational at present. Advancement of technologies related to low cost and efficient pretreatment methods as well as organism development were identified as critical requirement. Biohydrogen is still in its infancy due to challenges associated with distribution and storage. Also, supply of raw materials is unstable in some locations. Government policy is a major player here too. We need policies encouraging environmental stewardship. Hopefully soon scientists will be able to overcome this stagnancy and establish biofuels industry as viable alternatives to petroleum. Thank you everyone for watching.
Hello everyone, welcome and thank you for coming into my presentation today. My name is Dr. Irene Fakokun. I recently defended my PhD thesis at the University of Manitoba at the Department of Biosystems Engineering. Today I'll be talking to you about oleaginous yeast, a viable alternative for biodiesel production. Many of you have heard about biodiesel, even biodiesel production, but maybe not many of you have heard about oleaginous yeast. And don't worry, at the end of today's presentation, you'll be able to tell someone else a little bit about oleaginous yeast. So why do we even bother with this? There's a growing world population, and right now we're at 7.8 billion people, and that's still growing. And there are growing concerns over the long-term availability of fossil fuels. And we know that there are deteriorating environmental concerns born out of increased industrializations. We see this with the flooding around the world. We see it in the forest fires around the world. And there's so much concern about this. And we think about cleaner, more sustainable kind of energy as scientists. So we need to exploit this. Scientists came up with the first generation biofuels, and this involved the use of staple food, for fuel. However, there's a problem with this because it conflicts. There's a conflict between food and fuel. When you're using corn and things like sugarcane for fuel, you're robbing someone else of food. Also, there's the conflict of arable land. You're robbing the available arable land for the purpose of food, for fuel. So then scientists came up with the second generation and third generation biofuels, of which one of them is the oleaginous yeast. That's where it falls. So oleaginous really means that oil rich. That's what it means. So oleaginous yeast are, in theory, yeast that are able to store in their cell weight, in their dry cell weight, up to 20% as oils. And by oils, I mean lipids. And lipids in particular, I'm talking about triacyl glycerols. These triacyl glycerols can then be trans-esterified to biodiesel. So there are different types of oleaginous yeast. And I studied a particular one for my PhD program, which we called Rhodotorula diobovata. Very interesting yeast, because this yeast is even able to store up to 70% of its dry cell weight as oils that we're interested in, as triacyl glycerols to be in particular. And this is very interesting. And in addition to that, it's able to produce other bioproducts of interest, including carotenoids. Carotenoids are colorful substances, some of them that you find in your carrots. So that's not the subject for today. Today we're talking about the triacyl glycerol part of this yeast. So very interesting yeast. And why is this so spectacular? Is because this yeast have several advantages over other sources, and that includes the ease of culturing. They are very easy to grow. You can grow them anywhere in your lab. They have a unicellular structure. They are amenable to change. They have a high growth rate. You don't have to wait for months for it to grow. You don't have to wait for a year. So a couple of days, in fact under a week, and you see growth. They have a fast rate at which they can synthesize and accumulate lipids. So in a matter of 4-5 days, they're already making lots of lipids. And there's also low cost of media needed for this growth. And this is so important when we think about the economics of biodiesel and biofuels in general. We think about the cost. So with Rhodotorula deobovata, for example, we're even able to use waste materials, waste biodiesel products, as a source to feed them, and then they're even able to produce lipids that we're interested in. So that's fantastic, and this is what we need as scientists. From my work, I discovered that the culture conditions actually affect the cellular activity and lipid production in general. So when I talk about culture conditions, I'm talking about the carbon source. What kind of carbon are we feeding it? So are we feeding it with glucose, glycerol, or corn stove, or whatever? We're also looking at the carbon to nitrogen ratio, how much carbon to nitrogen is there within the system. That also influences how it grows, the nitrogen sources as well, the minerals and the inorganic salts. So this is like the human being having balanced diet, the kind of diet that you feed yourself, how balanced the meal is, it actually affects how you grow, how you develop, and same as this yeast. So we have been able to find also its optimal temperature of growth and what kind of pH actually affects the growth. And then another work that I did was looking at the aeration rate and how much aeration that it needed to grow optimally, and this was very important. So from the work that I did, I was able to deduce that total biomass production was so much greater under aeration enhanced conditions, that it was important to aerate the environment and aerate the system in which the yeast was growing, because we were having, in fact, more tags, more triacylglycerols. I call them tags. We were having more triacylglycerol yields under aerated conditions. We were having other bioproducts also increasing more because we were having a higher biomass under these aerated conditions. Another thing that I found out from my work was that the overall substrate consumption was lower. So in this case, I'd used glucose. I'd fed the yeast with glucose, and I needed just a little glucose to make even more lipids when I aerated it compared to when I didn't aerate it as much, So these are some of the kind of work that we do in our lab with oleaginous yeast, in particular Rhodotorula deobovata. We had worked on other yeast as well, but for this presentation, I'm just saying to you some of the things that we did with the deobovata. Another interesting thing that I did was to look at the genome of the yeast, to sequence the genome of this yeast. And by genome, I mean all the genes in the yeast. And this is so important because it helps us to know what are the genes that are responsible really for producing the triacylglycerols that we're interested in and other bioproducts, and to see how we can enhance these genes, by maybe impacting one pathway over the other, or by maximizing both pathways of interest. So that's some of the work that we have done with Rhodotorula deobovata. So again, just to round up, this is just to show you that oleaginous yeast are viable alternatives. They are easy to grow, and they grow really quickly, they grow well, and they're able to produce as much lipids, even up to 70%, like in our case with deobovata, where they're able to produce and synthesize up to 70% of their dry cell weight as lipids of interest, and this is a really good alternative for biodiesel production. I hope that you guys have been able to learn a little bit about oleaginous yeast today, and you'll be able to tell someone else about some of the work that we do in our lab. Thank you very much for your time. Goodbye.
Hi, I'm Mariam Bofankar. I'm a master's student at the University of Winnipeg. I've been working on digital agriculture project for almost 2 years. Before explaining what I've done, I want to tell you a story about I was interested in the project. I've always asked myself how we can have much healthier and organic products when I was at supermarkets. I've seen that just a few shelves are contain healthy products, and most of them are non-organic ones, and fruits and foods are contained and covered by chemical things. You can see that, for example, when you buy apple, you can see the white spots on the apple. That means that it's covered with chemical things. And I was thinking that it would be great if I can do a work or a project to contribute a future agriculture that can produce much healthier and produce more organic foods. I was lucky that on my master's journey that I can find an opportunity to help producing healthy foods by using artificial intelligence. To answer why most products in supermarkets contains harmful chemical things, we need to review and compare the agriculture industry in the past and present time. As you can see in the picture, we had more healthier and organic foods. Why? Because farmers or gardeners work on the fields and care plants one by one. They moved across the fields of their farms, remove weeds around the crops, and also recognize the unhealthy leaves or damaged plants, and they cut it to produce healthy yield. And even we had healthy animal products because they were fed by origin plants. But by growing the population and getting high demands on agriculture foods, the traditional and the past methods of agriculture didn't work. Farmers had to make more yield with fewer waste. The farms was getting bigger and bigger, and the farmers could not care plants individually. So the chemical things came to agriculture industry. Currently, farmers use huge amount of harmful chemicals to prevent the products from insects and damages and keep products in a good shape. Currently, we can see farms, all products on the fields are pesticides by agriculture aircrafts that moves on the farms and they spray all the plants, all the weeds, crops, healthy or unhealthy crops to kill insects or weeds. So the healthy plants are covered with such an harmful chemical that causes cancers, respiratory diseases, and other harmful effects on human and even on the environment. Digital agriculture provides a solution to protect plants from insects and being damaged by curing plants one by one. So it would take advantage of past gardening methods in big farms to produce a big deal of healthy agricultural products. The project that I'm working on is building a robot to move across the field and act like a gardener in the past. You can see on the picture that a robot uh, can can walk on the field and it acts like a gardener. It has like an eye and it has a mind to distinguish between crops and weeds. Actually, my research is designing the robot's brain, the brains that can distinguish between crops and weeds and also determine the health condition of plants on the field. I've worked more than million number of plant label images which are important for Canadian agriculture industry like canola plants, soybean plants and other famous plants. After designing an application in purpose of data pre-processing and data pipelining to remove noise data and organize plant label images, I started to design several machine learning architectures and obtune them. One of the strongest deep learning model I designed that specifically work on the big data sets called generalists and specialties is the name that I this is actually the name that came from mine. I mean, it's a specific name. The generalist and specialties model has 98% accuracy. It means that among 3,000 test plant images, which are very, very similar to each other, some images that the farmers or gardeners are hardly can recognize that is it weed or is it the crop, but the algorithm can recognize it by 98% accuracy. It means that it can recognize it by 98% images are distinguished correctly. My algorithms and other researchers' ML architectures can bring a big revolutionary on agriculture that enables autonomous farmer machines move across fields of farms and distinguish weed and crops and determine health condition of each plant. In this case, without using pesticide or herbicides, the weeds are removed and unhealthy leaves of the plants are cut by robots. So hopefully in future, all the shelves our supermarket will filled by origin products. And I hope you find it interesting, and I hope you some ideas bring to your mind about digital agriculture and using artificial intelligence in this field. And if you're interested in knowing more about the project, the research details, please feel free to contact me and I can share with you the model that I work, the detail about research, and I can talk with you about the technical things, more technical things. Thanks for your time, and thanks for Manisha and Nisha to organizing Soapbox Science. I hope I can have more talk on this event. Have a good time.
Hi, my name is Kelsey Burke and I recently graduated from the University of Delaware in the United States with my PhD in biomechanics. My research is always focused on concussion, so I'm going to give you a little background on concussions because they are such a hot topic. So a concussion is a mild brain injury. We can classify a brain injury as mild, moderate, or severe, but a concussion is on the more mild end. Now, you may have heard the word concussion. Maybe you've experienced one yourself, or you know someone who's experienced it. Maybe you saw the movie Concussion recently. Either way, chances are you've probably heard of a concussion. So these mild brain injuries can result in a whole different bunch of symptoms, such as headache, dizziness, nausea, light or noise sensitivity. And these are the symptoms that are typically going to be assessed when you go see a doctor. However, we do have some other tests that look at how your brain functions, how quick you are to react to something, what's called reaction time. We can assess things like how your eyes and brain talk to each other, different clinical assessments. None of these assessments or tests are diagnostic, meaning they can't diagnose a concussion, but they can help your doctor determine whether you have one or not. A typical recovery from a concussion is about 7 to 10 days long, and some people will experience symptoms longer than that or persistent symptoms. Now, we've had a lot of research on concussions that typically happen in those 7 to 14 days and sort of what's going on with your brain during that time. However, the new area of research is what's going on in people who don't recover in that normal time frame. So most of the concussion research over the last number of years has come from a younger population, such as high school kids, college-age student athletes. However, the literature or the research on adults, anyone sort of over the age of 25, has really been lacking. There's not much out there. And so I had a big interest in what happens when symptoms don't go away in adults. Adults have a job, they might have to drive to and from that job, they may have kids or pets to take care of. gotta go get the groceries, things like that. Very, very normal daily activities that when they have symptoms that aren't going away from their brain injury, they can't do them, or they become really hard for that person to do. So I wanted to look at what was going on with these people and can we help them? Can we figure out a way to maybe help them get better? So I looked at people who had had persistent symptoms, meaning 3 or more months since their concussion. I compared these people to healthy people who did not have a concussion, and I assessed their symptoms, their memory, concentration, reaction time, and how well their eyes and brain talk to each other throughout different assessments. So I'm going to demonstrate one of them for you. One of them is called the VOMS, and so this is a way to get your head and eyes moving independently and together, and to see what goes on. So the first one is, I'll move my finger up and down, and you have to follow it with your eyes. Don't move your head, just back and forth like that. The next one, I put my fingers out like this, a little wider, but I'm doing this so you can see. And I have the person just look back and forth with their eyes between my fingers without moving their head from side to side, and then we do it up and down. After this, I take a popsicle stick that has a T on it, and I bring it in towards their nose, asking them to stay focused on that T. When that T splits and you get sort of that double vision, like when you try and cross your eyes, the T will look like there's 2 T's. So I ask the person to tell me when they see that, and then I stop the popsicle stick and I measure how far that popsicle stick is from their nose. The next one we do, you stick your thumb out like this, you stay focused on your thumbnail, and you shake your head back and forth like this. The next one after that is the same thing but sideways. So you flip your thumb to the side and you look up and down like this. The last one, you still keep your thumb out like this, staring at your fingernail again, but this time you're going to turn your upper body to the left, and then you're going to go to the right, and you're going to keep going back and forth like this until I say stop. Now what happens is you and I, normal healthy people, should be able to get through all of these movements without really having any issues. No symptoms, we don't get a headache or anything like that. However, what we noticed was that people with concussions or persistent issues from their concussion really struggled to get through this whole assessment and all of those movements I just showed you. So we found some interesting things using all of those clinical assessments. That suggests that maybe we can start using some of these assessments in the clinic where doctors and other types of healthcare providers are making the diagnosis of a concussion. Additionally, I mentioned that my PhD was in biomechanics. So the biomechanics side of things was just looking at how the body moves, but also looking at how the brain works. So what I did is I brought people, the same 2 groups of people, into a motion capture camera lab. Motion capture cameras are the same type of technology that are used to create video games. So if you've ever seen that, you've probably seen someone with a bunch of silver reflective dots all over their body. And then when they move, a computer is able to assess how they're moving, how quickly they're moving, their different joint angles. So if they go from here to here, they can tell you how far that was, things like that. So what I did was I brought people in, put those silver reflective markers on them, and I had them walk and think at the same time. And while they were thinking, I was asking them to respond to something, some sort of test. So for example, I would ask them to spell some words backwards, or subtract by a specific number, or list all the months in reverse order. And so what we found was that the people who had persistent concussion symptoms were really not good at doing this task of walking and thinking and talking. We called it a dual task. And so what happened was, a lot of the times people were okay at answering my questions. They did them accurately, they got them right, but their balance was off. And that's what we were able to assess with these motion capture cameras. And so their balance wasn't as good as the healthy controls, even though everyone was answering pretty accurately on these thinking tests. And so basically, this tells us that people people with persistent symptoms might struggle to do two things at once. Now, why do we care about this? Well, think about walking down a hall, maybe to go see your friend, to go, to the next classroom, to go to an office. You're probably also talking to someone. Maybe you're also looking at your phone. You're doing so at the same time that you're walking to wherever you're going, you're doing something else. You're thinking, you're talking. Dual tasking is a very typical thing that we do on a daily basis, and most of us healthy people don't really think twice about it. But those with the persistent concussion symptoms, they really struggle. So when they walk down the hall to the classroom, to an office, they go grocery shopping, they likely aren't as good at doing those things, but also talking to someone or also being on their phone. They might have to just strictly focus on walking down that hallway. And so this is a very realistic, everyday implication or reason to do this research. So I'm hoping that my research will really be able to help people like doctors, physiotherapists, occupational therapists, people who help those with persistent concussion symptoms get better. And the way we could potentially do this is by including dual tasking into their therapies. So getting them to not just walk, not just think, but putting them together, making them do those two things at once and really get their brain going while they're physically moving. Hopefully, including that in their therapy will bring them back to normal. So thank you for listening, and feel free to contact me with any questions, comments. I love talking about this stuff, and I hope to hear from you soon. Thanks.
Hello everyone, I'm here to share my research project titled Privacy Preserving Patient Data Publishing. The research is all about preserving the privacy of individual record under what we call temporal hierarchy. What do we mean by temporal hierarchy? Temporal hierarchy is like a bottom-up aggregations of record using time frequency starting from the second upward up to the year level. So there are different levels of hierarchy depending on trust and the level of granularity that we wish to release about a patient or about any form of data set in such a way that individual record within that data set is unknown, their privacy is preserved. Let's say, for instance, there are 100 people in the ICU. The information is 100 people in the ICU. Nothing else. There is nothing about individual record, when they arrive, how they arrive, their name, their SIN numbers, and not sort of things in the publishing of the data. So depending on the granularity, the level of trust to the person of the publishing platform, we can release data based on different hierarchy of temporal or different hierarchy of locations. So what we are trying to do is to collect different events, not necessarily hospital events, but different events, and find a way of connecting them using temporal or the spatial relationship, while at the same time preserving the privacy of individuals within that record. Let's say for another example, let me share my screen with you. This is a typical example of our model that we have created. If, for instance, the information about this that we've collected contains second, minute, hour, time, the daytime, months, quarter, or year. These are different levels of hierarchy, and the location as well can be linked to this data set by preserving any level, by preserving up to a particular level. Let's say, for example, we remove every time aspect of this information. That means we are only reporting, or we are only publishing the record by day. And at the same time, we can go ahead and release lesser information about the location of the event. For instance, as I've said, if there are 100 people in the ICU, nobody is interested whether they're in St. Boniface or they are in Health Science Center. But we are saying there are 100 people in ICU on a particular day. We are not releasing the time of their arrival. We are not releasing how many people, the name of those people one by one, what is the agenda, all the information about individual or in that record are protected under what we call temporal hierarchy. And our temporal hierarchy allows us to move up or down. For instance, we can move up to the month level by aggregating up to the month level and aggregate the location to the city level from the street level, in such a way that if this information that we want to release, we observe that the third party that we are releasing the information for, the level of security or the level of trust that we can ascertain about these particular people is a monthly. We can release monthly data rather than releasing up to the time level data to this individual. So we keep it to the month data. Another option for us is to release information on a yearly basis rather than on a monthly basis. So the hierarchy gives us flexibility of preserving the information at a particular level of the hierarchy, and at the same time, we may keep we may pick a particular level of the hierarchy and varies the location as well, rather than saying there are 100 people in a store today, in a particular store today. We can say there are 1,000 people in our grocery store in Manitoba, and that is we are aggregating those information in a way that the individual content of those information is unknown to those who are using the data set. But at the same time, the main point of the data set is still valid. That is, if there are 100 people in the ICU, there are still 100 people in the ICU. That means the information is still valid, despite the fact that we are aggregating. The main information is that it still remains valid, that there are 100 people in the ICU. So the protection of those information is based on the temporal hierarchy or the location hierarchy, or combination of both. So we can say that we have 100 people in the ICU. Yes, we have 100 people in the ICU, but at the same time, we can come down to the level of time by saying at 10 o'clock, there are 10 people in the ICU. That means out of that 100, we've released a particular information to another set of, probably another third party that needed that the time is very, very crucial to. And we are aware that their level of security, they've signed confidentiality agreements with us, and they cannot release that information that at 10 o'clock we have 10 people in the ICU. In other words, based on the level of trust, the level of security, or the level of confidentiality, we can vary the level of information that we release to individual, third party, or generally that we use in analysis of data. And that is what this research is all about. And it's not only about hospital. We've applied this model, our temporal hierarchy model. We've applied it to parking data sets. We've applied it to transport data sets, and we are still planning to apply it to various other data sets that have time series data. That is, they have the time and they can be aggregated under our temporal hierarchy model. That is what this research is all about. If you have any question, feel free to ask. Thank you.